Welcome to our very first Microphysiological Systems World Summit. It's really wonderful to see all of you here today and be able to finally interact with each other, meeting in person in the beautiful city of New Orleans. Are you able to hear me well also at the end? Okay. Thank you so much for joining our today's roundtable, which is entitled Developers Meet Regulators and End Users. My name is Magdalena Cassandra, and together with Annie Moison, we'll be moderating this exciting discussion. As you all realize, in just a few minutes, we have assembled a wonderful and amazing group of panelists for you today who join us in New Orleans from all over the world. They come from United States, from Europe, and from Asia, and they are representing all of the major stakeholders in the MPS field. We have today at the roundtable developers of various MPS technologies. We have end users from academia and industry, and we have some members of the MTS community who bring the regulatory perspective from various continents. Our goal today is to discuss the current progress and remaining challenges in implementation of new alternative methods to replace, reduce, and refine animal testing. But really, what we want to do today is whatever your role in the MPS field is, that you will come away with something, a new idea, a fresh perspective, and new strategy that you could apply to become more successful in your role in the MPS technology. So here is a format to give you an idea of what to expect. First, we'll take just a few moments for each of the panelists to briefly introduce themselves and tell us a little bit about their unique experience with microphysiological systems. Then we'll kick off the conversation. We'll ask just a few questions that draw out some major themes and trends that we should all be thinking about. And then at the very end of our today's time, we'll open up the floor to all of you to give you an opportunity to ask the questions that you were thinking about that maybe will help you to navigate upcoming opportunities and challenges in MPS field. Sounds good? Anyone has any questions? No? Great. So let's start with the panel introductions. Maybe we're gonna start from the right side of the table, Kaoru.
Hi, everybody. I'm Kaoru Sato from the National Institute of Health Sciences. This institute is a research center of Ministry of Health, Labor and Welfare, Japan. I've been involved in the project for regulatory acceptance of MPS and also in the project to develop blood-brain barrier MPS. Originally, I am a neuroscientist and the lab head of neuropharmacology. Thank you.
Yeah, hi everyone. My name is Adrian Roth. I work for Roche Basel in Switzerland. My current role is I'm Principal Director of Personalized Healthcare Safety of Pharma Clinical Development. Relevant experience in the field is mainly going back to my role before moving into clinical development. For 15 years, I've been working for Roche's preclinical safety organization, leading a team there in charge of cell models supporting drug candidate selection and characterization. And so as part of this roles, we looked into a number of different MPS systems and refined them, applied them. And actually also for my current role in clinical development, we're very interested in exploring ways how we could support personalized healthcare approaches using MPS.
Hi, everyone. I'm Tony Bahinsky. I'm currently the Chief Technology Officer for Vivadyne, and we're developing end-to-end automated high-throughput systems for 3D tissue chips. Prior to that previous experience with MPS, I was at the Wyss Institute for about 6.5 years, helped develop a lot of technologies there, and was part of the advanced technology team that led projects with DARPA and the FDA. Also have 25 years of experience in the pharmaceutical industry. Most recently, prior to joining Vivadyne, I was with GlaxoSmithKline, where I was the global head of safety pharmacology, and we've tried to, as Adrian, integrate some of these systems into the safety side of pharmaceutical development.
So Susie Fitzpatrick, U.S. Food and Drug Administration. So 11 years ago, I worked with DARPA and NCATS to start this program on organs on a chip, never imagining how it would blossom and all of the really wonderful science that came out of this this little program that we started and the awards we gave to several of you guys, including the VEAS and MIT. Currently, I chair the FDA agency's alternative methods work group. I also am the chief toxicologist or regulator at the U.S. Food and Drug Administration Foods Program that are also looking at the use of chips for chemicals as well as drugs. And I'm excited to be here today and honored to be on this panel with all of my co-workers, co-scientists. So thank you. And very interested in what you want to tell the FDA. So.
Good morning. My name is Jan Lichtenberg. I'm the co-founder of Inspiro in Switzerland. I'm a microfluidics engineer by training, but moved into life science many years ago. We started Inspiro in 2009 with the idea to bring scalable organ-on-chip technology and scalable organoids and spheroids to the market to help improving drug discovery and safety applications. Our passion is kind of finding that balance between biological relevance on the one hand, but also then integratability, automation compatibility for these systems to be able to really transform the way how we work with these devices and to be able to replace a lot of the current 2D and cell suspension culture with more relevant 3D models.
Hi, Nina Hobby from Albelix. I'm co-founder and co-CEO for this company. It's Swiss-based, it's a young company. And my background is in medical physiology and lung physiology. And as a PhD student, I always worked on advanced in vitro systems. And for me, it's great now to see that organs on chip and MPS has a real field that people making impact and that we really can do something for the future altogether.
Hello, everybody. I'm Danny Levner. I'm the Chief Technology Officer and one of the co-founders at Emulate. Like many of my colleagues, my roots are also in academia back at the VEAS Institute. But for the past 7 or 8 years, we've really been focusing at Emulate on bringing this technology commercially to the world out there. So I'm happy to speak about that as part of the panel.
Hi, my name is Q Beck, CEO of the Curator. As you may notice that I'm a last-minute sub for the newly. He's supposed to sit here as a panel today, but he somehow missed his flight. I hope that he can find a way joining the rest of the meetings. Anyway, the Curator is a spun-off from the Nulis Lab in South Korea, and then headquartered in San Diego, California. We mainly focus on developing high-throughput MPS platforms for novel disease models in areas of the high unmet medical needs. Before joining the Curator, I used to work at the Vertex Pharmaceutical for various drug discovery programs. Thank you.
Excellent. Thanks, panelists, for the self-introduction. So we are ready to kick off this panel discussion. We have 3 main questions. I will raise a question, panelists will answer, discuss, and then it's open to you, audience, at the end. First question, are we ready to replace regulatory required animal studies with MPS? And I direct this first question to Tony, Adrian, given your experience interfacing pharma and regulators. So what do you think about that? Are we ready to replace animal studies with MPS?
Thanks for handing off that really simple question. I think we're close. I think there'll be on a case-by-case basis and depending on the context of use for the study. I think we're closer on the efficacy side, especially where there are disease models that are not human relevant. I think MPS can play a significant role there. Biologics, antibody studies where there are not relevant animal models. Again, I think MPS can replace those and actually be used in regulatory packages. On the safety side, I think we're going to need to be a little more conservative. I think there'll be ancillary data initially, and then potentially, as they show their translatability to the clinic, they may replace, certain studies, for certain liver toxicities. But I don't know, Adrian, what do you think?
Do you agree with that, Adrian?
Yeah, no, thank you very much. I fully agree, and would like to add that. Close is very optimistic if you really want to completely get rid of animal tests, particularly for drug development. But I think there are certain areas where we will step by step replace animal tests. I would like also to mention that in the end, what counts for drug development is of course patient benefit, and if MPS can show that we're generating more safe and efficacious drugs, then they will replace current tests, if be it animal tests or other types of tests. And so for some areas, I think we see nice success cases, and for others, there's still a little bit more work ongoing. I see particular opportunities for some of the newer modalities where maybe the spectrum of side effects to be expected is already a bit more narrow than for off-target effects with small molecules. So I see opportunities there that animal tests can be already at this point replaced with MPS models.
Yeah, thank you. Daniel, you want to comment?
Look, let me challenge the question a little bit. I think for one, let me divide the world between efficacy and safety models. With efficacy in development of biologics, there is certainly a lack of animal models, and there is a great need for organ chip models or MPS models. And maybe the question there isn't even replacing, because the good models don't even exist in the animal world. But focusing on safety, I'm not sure that we should be asking ourselves, when should we replace? Because it's actually an inflammatory question for many. For many in the regulatory world, for example, you're taking away this method has been trusted for decades, and you're taking that away from them. And that's not necessary, because the more data points we have, the more information we have, the better decisions we can make. Just like we don't make decisions with a single species of animals, we look at least 2 different species of animals. So too, we can add chips to this set of information to get a more complete picture. Now, with time, as we gather more information, more drugs, more confident system, things will happen, things will adjust. We'll refine and reduce the reuse of animals. Maybe eventually we'll get rid of one of the species. Maybe eventually we'll get rid of all the species. But the question of, are we ready to replace now, honestly causes an antagonism that's not necessary. We can coexist for the time being.
Thank you,
yes,
Daniel.
Daniel, I fully agree there, because I really think the history of animal testing is so long, it would be now not fair to say we have to replace anything in 10 years. So we really have to build the trust. And it's great that we have a big push to replace. This is really needed, but we also have to take the time and maybe not already compare. Just saying, making a lot of data, store them, evaluate them, take the AI, and then go for a next step.
Yeah, that's one of side of the coins. Suzanne, could you please provide us with regulatory perspective?
Well, I wish we were done with animal testing, but I think we're working on it. There are no requirements at FDA that you have to use an animal test, and we're certainly open to looking at alternatives and talking to our stakeholders about alternatives. And in addition to being open to that and working with people like you, people with CAT, to look at alternatives, we're also making sure that our regulators are up to date on seeing these in an application. We have a lot of chips from different developers in our laboratories. We're trying to learn as much as possible about these chips. There are a lot of endpoints that we're interested in that do not have good animal studies. And so I think we're looking at some of developmental, neurodevelopmental, immuno as maybe test cases, but we're working on it. And we certainly feel like you that we think MPS can give us very valuable information as we put safe and effective products on the market, which is our main goal. And hopefully we're getting there. So
yeah, thanks. And Jan, you wanted to comment?
So I think from my perspective, the approach angle, and this is a bit along the lines what Nina and Daniel said, is First of all, finding unmet needs in the industry that are currently not properly addressed with animal testing. So, Adrian, you mentioned novel modalities, bispecific antibodies, recombinant proteins, and so on. I think these are areas where animal testing, I think, rather poses a problem than an advantage. And I think that's where human-based models can really do a fantastic job. Another topic is patient diversity. And again, Adrian, you're looking at precision medicine and personalized medicine. I think we really need to encompass the diversity, ethnicities, but also different disease states more into our testing paradigms, not just for efficacy, but also for safety. And again, I think this is not really possible in the animal, but this needs scalable human models. What we, I think, for quite some while will not be able to deliver a systemic response prediction, because this is where animals are just the better solution at this point, unfortunately.
Yeah, actually, I joined this meeting as a regulatory person. So actually, I generally agree MPS is very good model, but in terms of regulatory acceptance, we have to collect much in-house data before that. If we could collect much data, enough data, the pros, a huge pros for MPS is we can study about the mechanism of the underlying mechanisms, and also we can use human specimens here. So, yeah, but before regulatory acceptance, we have to collect much data from the in-house data in pharmaceutical companies.
So are you receiving this data? Do you have data to analyze, to review sufficiently, or is it something you would like to see more from a regulatory perspective?
Not yet. Not yet.
Not yet, not enough.
Actually, in Japan, an MPS project has now launched. So we are now searching for what is a COU concept of use. Do a pharmaceutical company have?
Yeah.
So yeah, maybe you guys have many good examples for that. So please let me know.
Yeah, I mean, that's a good question to ask. What is the general feeling about? Do we have now data moving, to the preclinic and being submitted to regulators, or are we stuck somewhere?
So,
so if I go first, yeah, I think it's coming. There's certainly not a lot of examples there. However, I think regarding this discussion about regulatory acceptance, I think what we should remind ourselves is that there's only relatively few really regulatory required tests that need to be done in order to file in a new drug application, and those will be done. But that doesn't mean that everything else is not important. There's a whole lot of tests and experiments are being conducted, which in the end are not part of the regulatory documentation, but they are absolutely key for us internally to take decisions on molecules to move forward or not. So I think we should not be completely obsessed with the regulatory acceptance in terms of an MPS test is only valid if it has the status of a HERG assay or something like that, because that's a matter of maybe 20 years or so of validation.
So for sake of time, we take one last comment. Q.
Yes. So in the last 10 to 20 years, I know that it sounds a bit off track, but the last 10, 20 years, there have been a very successful case of the drug discovery, the programs that made no use of the animal models for the efficacy and the potency, like the cystic fibrosis 3 drugs. And also, in this morning, they announced that some of the data for that pain program for the NAB 1.8, they didn't use the animal models for the efficacy and the potency at all. So I think with a specific understanding of the disease model, disease biology, and also the judicious selection of the target. And if we are using that some near the physiological the model, the MPS driven, I think maybe that we do not need the animal model down the road for that.
I think hearing more about case studies, like examples from all of you. We don't have time, unfortunately, this morning, but perhaps in the coming days. But that'll be very insightful, I think, for us all. And it's time to switch question. No?
Yeah, now we're gonna focus on the challenges, and I would like Jan to take up the first question or the second question or the roundtable. How can we clear the main obstacles to wider adoption of microphysiological systems?
Mm-hmm. Thanks, because this is, of course, something that I think drives the whole developer community a lot, right? So there is a clear need for better model technology, but I would still say that the MPS systems, independent of whether it's spheroids or full-blown organ-on-chip systems, they still kind of scratch on the surface. And I think there are a couple of reasons. I don't believe that regulatory acceptance is really the key here, because the driving factors in the industry are reducing attrition costs, being more efficient in developing new drugs, maybe addressing specific therapeutic areas that are currently not covered by therapeutic paradigms. We are working heavily in NASH, for instance, and there's really no drug on the market for this. So the important obstacle that we see is the lack of confidence yet in these new technologies, not just from a topic of biological relevance, but also how does it perform in the lab? What is the cost of ownership? Can we use this broadly? And there is a plethora of solutions, from very simple automation compatible solutions to quite complex ones where you need dedicated devices. And I think we as developers, we need to be we need to work very closely with end users to make sure that the advantages are understood and that the context of use is understood, because not every system is equally useful for any application in the drug development chain.
Thank you for that. Nina, could you speak to the challenges of your own company?
Of course, happy to do so. So I fully see also we really have to take the risk to go very early already to the end user and establish something that they really then can use also for applications. So in our company, we really have the benefit that anything that we do is mostly in the preclinical phase. This means that it's very exposed. You get a lot of feedback, what is working, what is used. And what I would hope for is also that maybe it's somewhere tracked if, for example, an end user used MPS in the application, somehow that you can make a cross during application, that it's easy then for the regulatory to see who used it, who doesn't use it, and also got the feedback, what was working and what not. And also other end users can then use the same process. Also in regarding of price, I think this is a big limitation still in MPS. As more it is used, as cheaper it will get for the developer, so we can offer it to a better price. So anything that is validated and accepted, we know we can do it next time the same. We can make better prices for the next customer or for the next end user. And the third point, which I think it's personally very important, is also to give access to students. So with big core centers, that already in the education, people know that MPS is something which they can use later than in their career.
So how are we going to support that? Do we need philanthropy?
How we support this? Yes, I would really, this is very, very visionary, I would say, maybe also a budget price-wise. But I think it would be really good when using MPS, because currently it's volunteering. People use MPS on top because they have no other option, or they think, okay, we start with the MPS on top with the animal testing, so that they can somehow reimburse part of these costs. Because this, of course, would foster that people say, no, it's not on top. I have more data for the same price. And for the education, of course, we should have facility centers.
Thank you.
Yes. I think there is no single silver bullet to overcome all the obstacles, because the different beneficiary group of the MPS is facing the different obstacles. So I think in general, the first most important thing I believe for the pharmaceutical company is to provide that the MPS very compatible with that the high throughput, the comparability, the instrument and liquid handler and the high throughput screening system as well. And also that's addressable by the very standard instruments. I think that is one of the key things. Then there is another issue of the robustness, because that usually pharmaceutical company cannot tolerate that sort of the failure rate that is okay in the academic settings. So in short, I think it is very important to provide that high throughput compatible and also the robust enough to be widely used.
Maybe we can get the take from pharma. Do you agree?
Yeah, sure. I think the robustness is a key one there. So knowing that there's good translatability to the patient, if you can get biomarkers and endpoints that translate directly to the clinic, I think that's really important. Or you can have surrogate endpoints that's important also. But I think the ones that are really holding it back, especially from pharma, are the scalability. We're still very low throughput on a lot of these systems that are available commercially or even in the academic environment. I think you really need to scale it up and actually be able to do tons of experiments and different types of variations on the cells, the conditions to get optimized systems too. And the systems now are just too slow. Reproducibility is a big thing also. You want to make sure that somebody in the same lab that's doing the same procedure can recreate that same experiment and the same tissue type and get the same endpoint and results. Even more important, we can't even do that really reliably, let alone at another site or in another country across the ocean. So I think that's really key too. Automation can be a key to both of those things. One, to help with the scalability, and then two, with the reproduction. If you can automate a lot of the tissue culturing and production of the tissues, I think you get that, remove some of that human component of human error from the systems.
So Jan, you are on the board of Society for Lab Automation.
Yeah.
Can we hear from you? Should we be developing additional instruments or make them adaptable?
I think this has been a big topic at SLAS. How can we automate this? Because ultimately, you're totally right. For a lot of applications, this needs to be throughput compatible. Also, if you want to use the same models from, let's say, secondary screening to target validation to further investigation, if you switch systems, this always introduces certain question marks, which are not necessarily helpful in the process. So if you want to pull this through, you need solutions that are robust and scalable. And I think this is a general situation that we have in our domain. Everybody knows the hype cycle curve, right? So I think we are a bit past this in the organ on chip and MPS area. There was maybe too much promise and then a certain under delivery on the results, because ultimately a non-scalable system that creates a great result is kind of leading us a little bit into a dead end. We always have to keep the industry interest in mind to create something that works 24/7 in an automated lab environment. And I think this is what we've seen also in our industry from a developer perspective, moving more into under-promising and over-delivering than the other way around.
Do you think we can catalyze wider adoption by making those models perhaps more affordable for a wider community?
Personally, I think, and I agree with Nina, so scalability also comes, or price goes down with economies of scale if the technology permits this. And so a wider use of the technology is definitely going to help not only the supplier of a specific solution, but also other suppliers, because in this case, the flood raises, I think, all boats. And I think this creates a benefit for everybody. I think what we could use from a regulatory perspective here is help in terms of developing, maybe not validation, because this is already a very high bar, but qualification requirements. How should we characterize the model in terms of its cellular composition, in terms of the mechanisms, biological mechanisms that are built into that model? What are compound sets that we can use for benchmarking? not to single out a specific vendor, but for giving everybody a certain peace of mind that a certain technology lives up to a standard that is required. And I think that could help us a lot.
Want to comment on that, perhaps, Kaoru or Suzanne from regulators on this wider adoption? What do you think could help smaller companies perhaps also to?
I agree. I mean, I think I don't see any regulatory barriers to FDA accepting an alternative method, MPS or otherwise, if we have confidence in its context of use. And I think one thing FDA wants to do, we are looking at how you decide relevance in that, and that's something I think we have tried to do in partnership with our stakeholders, because we all have the same goals in mind. So it's not just our question, or not, it's where we all work together. We're working a lot now with the IQ Consortium MPS to talk about standards. We invite any people with new methods to come in and present them to the FDA through our program, like once a month, and occasionally we'll adopt one of those and bring it into our laboratory, all in an effort to really understand, like you, what we need to move these forward to get more, because we want more predictive tools.
So that brings us very well to the third question, Suzanne.
Pardon?
That brings us very well to the third question, if I may switch to the.
Okay.
No, that's not what I'm saying. Just for the audience and sake of time, I'll just like verbalize the third question, which is very relevant to what you're saying. So where do you see opportunity for collaboration to support MPS development, adoption, and acceptance? So you can continue.
Going.
Yes. No.
I see working together. When I started at FDA, we were on one side of the table and everyone else was on the other. It's not like that anymore. We all want the same thing. I think it's not just us we're not actually, I don't think the regulatory block, it's the industry being worried about putting new technologies in and what are we going to say about them? But I think that means we just need, like this meeting here that Kat has organized and that Dan is, this is where we all talk together about what we see, how do we show relevance? How do we show robustness? Give us your ideas, because we're working on a lot of things and we want to hear from those people working on it. We have chips in our lab trying to learn this too. So I think it should be considered a partnership to move these new, exciting methods, which we helped start. And so we are invested in at FDA, move them into good tools to put better products on the market. And ideas, we have a website, we have alternatives at fda.hhs.gov. Send in your ideas, send in anything, proposals for workshops if you think it'll advance the field. We are very open to doing this.
So Jan and then Kaoru.
Just maybe quickly, and I fully agree, like iStand, for instance, is a great initiative in that direction. And I think we have a good example that we started last year. Our interest was in reducing, not replacing, but reducing animal testing in the safety space. And so we developed together with partners a set of liver microtissues from different relevant species in the preclinical space so that we can test species-specific tox effects early on. And instead of doing this just inside Insphere, we decided to team up with a lot of pharmaceutical companies like Genentech, AbbVie, Sanofi, and many more that are all chipping in, that provide compounds that had species-specific toxicity, but not in man. And these are exactly the examples that we're interested in. And so we brought in academics to help us in that project. And we also then sent our feelers out to FDA using iStand so that we can really create kind of the full package, the information that's necessary, but also get the regulatory guidance early on. And so this has been so far a great experience for us.
Kaoru, I'd like to hear you from the Japanese perspective. Huh?
Yeah, actually, I've been concerning what kind of regulation is suitable for MPS. I mean, MPS is comprised of many component technologies. I mean, the cells and device itself. So what kind of regulation is the shortest cut to accept for acceptance for everyone, acceptance by everyone. So I mean, for example, MPS package. I mean, MPS test itself, regulation is appropriate, or device itself or cell itself, and which is better for the shortest. I'm not sure at present, but if you have any opinions, please let me know.
And do you see a good collaboration between maybe like the developers, users, and pushing that to regulations? How is the collaborative landscape over there?
I don't know. It's I mean, I'm talking about the quality of cells and devices, and also the MPS. The resulting MPS. The quality of the MPS test is enough to approve the new drug. I mean, this is my core question. I mean,
maybe we could learn from.
cells and device itself is very.
That's an effort. Should be a collective effort
to get the robust results. So,
okay, maybe we could learn from the FDA of how to build the partnerships of this type. Suzanne, could you please comment how to build the partnerships of this type between all of the stakeholders to make it really work? Suzanne,
I'm not quite sure what the question is, but I have a question for the
So
panel.
how to build the frameworks of this type that connects all of the stakeholders together? You have built multiple, right, with DARPA and CATS.
By having some money, obviously. We brought all the stakeholders together, didn't we? So, I want to know what the panel thinks. Do you need animal chips to show concordance to human chips to show concordance to humans? That is a debate that we have going on, and I'm just curious about what everybody here thinks about that.
Yeah, Nina and others from your side in collaboration.
I mean, is that how you're going to get them into regulatory use? I mean, I have feelings on it, but.
Yeah, I think Nina just wanted to comment.
It's okay, Adrian can start.
No fight, but ladies first, then we go to
It's a hard one.
Ladies first?
Ladies first.
Okay. No, it's an important question. I think there is a lot of pressure in this direction, and I'm actually not really a supporter in this. I really think we have a lot of gaps to solve. So the first step should be what we cannot do in animals and work on this, because then this part will be really nicely settled with MPS studies. And then later, when we have the history in MPS, that we then maybe start to compare. But currently we have so many gaps to solve, which is an animal testing really don't help us, that we focus first on that.
Adrian?
So, yeah, I do see a number of use cases where it makes a lot of sense to have also animal chips, if it's about, showing or absence or relevance for human, if there has been a finding somewhere in a preclinical animal test, and so these kind of exercise. But generally, I don't think it should be the general path to validate these kinds of models. It can help to validate the approach or the technology as such, because animal data tends to be more standardized. You have a lot of data to benchmark with, but I wouldn't go that route for every type of assay and application. If there's solid, good human data around, go for that.
Jan?
Yes, I also wouldn't use or wouldn't create an animal system just for the sake of showing the relevance of a specific technology, because at the end of the day, also the cell isolation and the cell source plays such an important role. Ultimately, it's about predicting clinical outcomes, right? That's what we're all interested. But we see that animal tests can be a substantial problem for pharmaceutical developers, right? If you have a signal in a specific species, it typically sets back your program by half a year, right? So this can be really expensive. And so being able to assess these risks early on and maybe already start to look into mitigation strategies before you go into an animal. I think it's a perfect reduction strategy of animal testing, but at the same time, it saves everybody a lot of money and especially time to get this drug to the market. And that's kind of where our interest comes from.
Nina.
Yeah, here a little bit an add-on also, because making an animal on the MPS is a lot of work, but you still have the whole data that anyway comes from the animal testings, which has currently to be done. And there, for example, we see a lot of alignment. So you still would have the feedback that your human MPS gives the same signals than the animal MPS, but it's not needed that you establish everything in the chip.
Could the real world data and AI help? Tony, what do you think?
I think we bandy about AI as the cure-all for everything, and I'd be very cautious with that. It can help. I mean, you need to first generate high-quality data, right, to feed into the systems. You need to be able to do that at scale, because these are very data-hungry paradigms. And then, hopefully at the end, if you do the right modeling, either you want to call it machine learning, neural networks, AI, you can probably develop, certain fingerprints and understand, where you're progressing towards either an adverse effect or potential efficacy. But I'm always kind of a little cautious now. It's like we're kicking the can down the road. Oh, AI will solve everything for us, and I think we're being a little naive that way.
Quick comments for the AI is I think MPS provided a very complex data set. I think it is impossible to choose at the right parameter, down the road. If you see something that for that, to find that correlation between the clinical response. So I think AI definitely going to help for that to find that right parameter for that data analysis.
Good. So we need resources, collaboration, and resources should go to human-based system, not animal-based system. I think that's quite a strong message and alignment we have here. That's good news. And now we have time for Q&A.
Yeah, we can open up the floor.
Go ahead, Magda.
To all of the audience, but before we're gonna do it, we would like to take the temperature of the room and better understand who the audience is. So everyone who is now developing new human cell models, please raise your hand. Okay, great. Quite some of you. Who are the end users of the technology that is supplied by other developers?
End users, okay. That's good.
Right.
You can be both, no problem.
Yeah. Who is interested in regulatory perspective, who is on the way to actually approach the regulatory acceptance? Great, perfect.
Can I ask who's actually a regulator? They're shy. I've seen a few hands. Good. So we probably need to have more from regulatory agencies joining us in these meetings. That would be very, very helpful and interesting.
So anyone would like to kick it off with the first question? You can just walk up to the
Go to the mic
here in the middle.
If you can just tell your name, please, and ask your question. Thank you.
Christine Mummery from Leiden. I just wanted to follow up 2 points that were made. I think one of them was by Jan. One of them is, how would I know whether my model's any good? And one of the things you can do is perhaps see or define a set of regulatory or a set of compounds to which the model should respond. And you could base that response on knowing what happens in patients or in humans. So we've already done that for the heart. We have 12 compounds we call diagnostic for a model. That's a stem cell model of the heart, in fact. And I think Lorna Hewitt's done it for the liver. So that's probably the way we could go with some organs, at least define a diagnostic set of tools. And the other point is a somewhat sobering thought. We've done a meta-analysis, or we're still busy with it, of the literature. And whilst for organoids, about 10% of the total number of publications are of reviews, the rest are primary papers. For organs on chip, there are fewer papers, but a third of them are reviews. which means there's still more promise than there is actual hard data. And I think there are a lot of researchers here. I think if we're going to make the field successful, we're going to have to produce a lot more primary data to see whether it's actually going anywhere. So thanks.
I fully agree with the latter point, right? So more data is definitely necessary, but I also think more data sets. So I fully agree with you. There are a couple of compound sets that have been developed by the IQ Consortium and others as well that help us in benchmarking. But I think we need to agree on let's say, a more holistic view on requirements to assess these models. For me, this would be 4 topics. One would be cellular content, right? What is in the model? If you talk about liver, is it just hepatocytes, or are we looking at Kupffer cells? What's happening to the innate and other immune system, right? How we approach that? Do we need it? Is this optional? Second point would be readouts that are available for it. Third point would be what kind of biological mechanisms are visible in that system, right? How can you elucidate that there is a certain response to a specific challenge, and that is not necessarily a drug, it could also be something else. And then the last point, this is exactly what you say, what is a useful compound set and what is the proper benchmark against it? And that compound set should reflect different modalities of compounds, not just small molecules. And it should reflect the different mechanisms of toxicity, for instance, in the liver safety model. And I think if we could agree on that from an industry perspective, end users, developers, and ideally also regulators, I think it would really help us to clear up the situation.
If I could follow up on that, let me connect a few of the questions that were asked here about barriers, about collaborations, about the compound sense for qualification, and so on. This community has been working hard for the past 10 years, even more, to produce tons and tons of data, lots and lots of papers. We've mapped out how many papers are in the industry, and the numbers are staggering. And in terms of promise of the technology, we're there. I think everybody believes that there's a lot of promise here in terms of showing, use cases and examples and case studies. We're there. There's a lot in terms of science. But what we don't have yet is any clear sign of adoption. And I claim, my claim is that as soon as we have some very real adoption, we'll open the floodgates to everything that's happening here, right? We're still stuck in the science stage, and we need to get into the adoption stage. Now, I think that the qualification, those compound sets and so on, is one of the main elements that we need in order to drive that adoption, because we need to get into the shoes of the end users of the technology and convince them that the technology does not just what we think is interesting enough for a first author paper, but what they actually need to use on a day in, a day out basis, including the cost, including the throughput, including the reproducibility and repeatability, but most importantly, that we actually solve the problem that they're trying to solve. And that brings me to that collaboration question, which is we need to get together around this, because up until now, I think we've all been trying to show the breadth and the depth of technology in terms of science. If we can drive this adoption, it will really shift this industry.
We have a question here from James Hickman.
So, how you doing all? Probably know most of you up there. But Jay Hickman from Hespros. I wanted to talk about the regulatory aspects because most people don't understand this. There's a really big difference between safety and efficacy. Okay, and the bar for safety is going to be much, much higher than it is for efficacy. We've already supplied the efficacy data for a drug with Sanofi for an IND that has already enabled authorization of a clinical trial that's been ongoing since April of last year. So that's already been done. What was for efficacy? Efficacy, the bar is lower, okay? Safety, the bar is much higher because the FDA does not want people dying, okay? And this is and Susie, that's the main difference here is between safety and the efficacy. I mean, Jonathan Goldschmidt, who used to be head of rare diseases at FDA, got up at a meeting we had 4 years ago and said, look, as you already mentioned, Vertex, okay, that was the seminal moment at the FDA in terms of Vertex had gotten a clinical trial approved for 12 indications for cystic fibrosis, 12 mutations. Then he got 24 mutations, then approved with only in vitro data. He said, we've already established the precedent, okay? And so we've been following that at this point. And like I said, we've already got authorization of a clinical trial. Okay. And I'll show the data in my talk, which is right after this. Okay. But that's the big difference. It's safety versus efficacy. And too many times people mix them up together, whereas getting things for regulatory approval through with the efficacy is much lower bar than it is going to be for the safety.
Maybe
There's a comment on, well, comment and then I go question to Jason.
our panelists comment.
So, I think it's a great point, Jim, and the publication that you guys have on this, I think is also leading the way. We've contributed data for IND filings and for FDA responses as well. I think the problem is not so much the openness of the FDA to look at this data, right? And I think Susie says very often we are very open to this and can become a little bit angry when people put this in question, right? I think the FDA is open to it. I locate the issue more on the side of the pharmaceutical developers. So Jim is working with some that are very open to bringing this in. Not everybody is open to that. And there are some concerns of adding a new dimension to the data set. And that's something that we need to alleviate.
Yeah, hi. Yeah, Jason Nickett, I lead the CIVM group at GSK. I thought just to add to James's comment about past chair of the IQ MPS affiliate. So we've usually broken it up from a context of use into safety, ADME, and then pharmacology. So leaving efficacy out of it because of pharmacology, where we'll see it as what's the dose prediction. So there's another realm of breaking it up even further. So that's probably one area to think about when from a regulatory point of view. I think the other thing is from adoption point of view, from your comment, Daniel, is trying to demystify what MPS organoids are when bringing them into pharma. So it's working with the bench scientists to help them figure out how to use these systems in the day-to-day use of it in the lab, and also how to establish it with already established protocols. So that's something also tissue chip developers have to think about. So I'd be interested what's your thoughts on how you're going about doing that?
Daniel?
Well, let me start with the efficacy question. We totally agree that the thought process around efficacy and safety are different. And one of the you're right. There's a dichotomy, something that we need to wrap our head around, right? In efficacy, there's a lot less legacy. There are no good models for many models, especially when you're developing biologics. But on the other hand, efficacy models tend to be somewhat bespoke because every company, every group is working on different indications, and there's a chicken and the egg problem, which is you don't start a drug program around an indication you don't have a model for, right? So it really requires very intimate collaboration to get that company to start developing something around your bespoke model. Safety and ADME are much easier because everybody's doing the same questions, which is why we find it a lot easier to figure out what products are necessary when we think about it in terms of products and applications on that space. So this is a question I actually challenge this whole group. How do we get into efficacy where the problem is great, the low-hanging fruit is there, but there is the chicken and the egg problem? Perhaps it's about fostering relationships with those people that are in the right place, right at decision points where drug development programs are started, to challenge them to start adopting these models early.
We have the time for a very last question.
Yes, hi. Frank Borelli from the Humane Society. From a toxicological point of view, I've heard some of the concepts mentioned in passing, but not necessarily discussed in depth. Concepts such as validation, relevance, harmonization. In the past, we know that in order to gain regulatory acknowledgement, some of these programs will have to be coordinated, especially validation programs. I understand that the field is still in a developmental stage. However, I don't think it's too early to start thinking about harmonization and validation programs, which will require a concerted effort from many stakeholders. I'm wondering what your comments are from the panel. Thank you.
Anyone would like to take up this question? Harmonization, Tony?
And maybe Susie can elaborate on this afterwards, but when you submit something to the FDA, I mean, so there's several processes. One is the qualification process, right? So you have a standardized drug development tool, and that's one way, and that's the much longer road. But if you are a sponsor and you're bringing a program in, if you have good internal data for that validation of whatever that assay is for the endpoint and predictivity, whether it be efficacy or safety, the FDA will consider that and utilize that in the IND package. Right? It doesn't need to be a qualified drug development tool. And I think that's where we get confused here. We spend a lot of time and across the industry, I see all these consortia where, we're developing, and we still haven't made a lot of headway, because we haven't come to a consensus over the last 5 years on developing these paradigms and the parameters that we need for qualification. You don't really need it. I mean, you can actually go to the FDA if you've generated that internal data to validate your own assay and bring it forth. So,
But it's not public. It's
right.
It's
That's right.
not public. It belongs to the company, and
Right.
if they decide to share it, it's their decision. But yes, you can bring anything as part of your internal discussions to the FDA. But the data belongs to the company. It doesn't belong
So
to
on
us.
that, I think it's a wrap up, but definitely we should take the time while we are all here to continue on sharing transparency, lessons learned. So how do we move the field together? I think that we can do better, definitely to accelerate how we move the field. So I hope that this discussion planted ideas, give you some insights and motivation to continue working on this amazing field of MPS. Magda?
I'd like to thank to all of the panelists for all,
Thank
all
you
of
to the
your
panelists.
insights, and to all of you in the audience for listening to us today.